Hello students, this is Dr. Faizan Mirza. In this video, I am covering with you how the cell shape changes along the mitotic cell cycle to identify the stages of metaphase, anaphase, cytokinesis, etc. from the given photomicrograph, because the questions that they give are having grayscale images and they become a bit tricky. As well as I'll be discussing with you the changing shape of the chromosome, a bit overviewing that so you can attempt the questions where the chromosome shape is being addressed, so you know how to just relate to that if the question comes. So this before going to this question here, let's just recall in the cell cycle, in the mitotic cell cycle, we know the stages of mitosis. This is early prophase shown to you. In early prophase, this is what happens in the early prophase and late prophase. We know that the nuclear envelopes start to disintegrate. The details that already I've covered in detail enough on in my video in the mitotic cell cycle of this content. So if we move on and we go to the cell, how it looks like in prophase. So you will always identify the cell whether the nuclear envelope can be seen or it is seen to be disintegrated. That must be prophase. If the centrioles can be seen and the chromosomes can be seen with the chromatids aligned, the nucleolus is being disappeared, so that must be prophase. If the cell looks like where the chromosomes are lined up in the center and you can see poles on the in the cell and you won't see any kind of nuclear envelope, so this must be metaphase. Within the metaphase, the chromosome splits at the center, at the centromere, and although it doesn't move, but the movement begins. The movement just begins. The movement can be seen to be significantly there in anaphase, in which the centromere has moved closer to its centriole as these microtubules start to shorten. The spindle microtubules. Again, you can see the nuclear envelope is not there. This is anaphase. In telophase, you will see the cell will have two separate nucleus. You will see the nuclear envelopes are there. The chromosome will be having a single chromatid each. The spindle fiber can be seen to be broken down, and you can see cytokinesis occurring. So cytokinesis just follows. The cell membrane invaginates from here. A cleavage or a furrow forms. A ring of microtubules starts to just move in such a way that the cell splits, and the phospholipids of one cell and the phospholipids of the other cell, they join on the same cell. So these two cells, they separate from one another. When we are discussing the shape of the chromosome, the prophase and the metaphase chromosome, they both look in the same way. The centromere is there. You will see one centromere and you will see two chromatids. This is one chromatid and this is the other chromatid. At the end of each chromatid, you will see telomeres. So this is what a prophase or a metaphase chromosome looks like. And there will be DNA present in the histone protein. That's why the DNA is condensed in the form of chromosome. Whereas when we discuss anaphase chromosome, in anaphase chromosome, there will be a centrosome or a centriole, which is pulling the microtubules as they shorten. The center, this must be shown attached to the centromere, because this is the kinetochore protein present here where it attaches, and the chromosome must have a single chromatid. Now, if it's one chromatid, it will have two telomeres, one at each end. Keeping this in mind, let's come to this question. This is October, November 22, variant 23. The early development of animal involves division of zygote to and daughter cells by mitosis to form embryo consisting of genetically identical cells. Figure 1.1, figure 4.1 shows the cell at various stages. You can see these various stages. If we just go to the details of the various stages. B. In B, you can see this thing is there, and this seems to be separating, and this is you can see a dense structure here. This is one cell membrane surrounding the whole cell. You don't see two nuclei, you don't see two nuclear envelopes formed. So this actually tells you that the cell division is not yet complete. In C, you can see the cell is almost splitting. You can see the cell membrane will start to unfold. The furrow is forming here. The furrow will eventually close down, separating the two cells from one another. Again, you can see the thick structure of the chromosomes aligned at both ends. In A, you can see this is the cell membrane, and all the chromosomes, they are lined up in the center. The dense most structure are always the chromosome, and you can see the microtubules are radiating from the centrioles at both ends. So keeping this in mind, what these stages could be: cell at A and cell at B. Cell at A should be metaphase, cell at B should be anaphase. Why is this anaphase? Because the chromosomes, you can see they seem to have moved to the opposite poles. In A, the chromosomes are lined up in the center. Figure 4.1 show the microtubules in the cells are dividing describe the role of microtubules in mitosis. So microtubules form spindle that attach to the chromosome at the centromere. The chromosomes are aligned along the equator of the cell. The microtubule attaches to the kinetochore of the centromere. During metaphase, microtubules begin to shorten. The centromere splits in anaphase. Daughter chromatids are pulled apart to the centriole by further shortening of the microtubules. You need to include the term of shortening of the microtubules because they actually are asking about microtubules only. State what happens in cell C, shown in figure 4.1 till two new cells are formed. So you need to describe the process of cytokinesis. The chromosome start to decondense, nuclear envelope reforms, which is telophase, nucleoli reappear. Cytoplasm and the organelles are equally distributed in the both daughter cells. Cell membrane invaginates as it contractile proteins ring constricts. This allows two new cells to form at stage C as shown in figure 4.1. The cells produced developing a whitefish are genetically identical, identified and explained two events that occur during the cell cycle that lead to daughter cells being genetically identical. During interphase, we know the DNA replication occurs in the S phase. This allows each DNA molecule to make its own copy by semi-conservative replication. Using the complementary base pairing, both DNA molecules are held together at the centromere. We know that splits in the metaphase. The splitting of the centromere occurs in metaphase. This is a common mistake that the students do. Keep that in mind that the splitting occurs in metaphase and the chromosome they start to move away from the chromatids start to move away from one another in anaphase and they move to closer to the centriole. This allows the daughter cells to have one copy of each DNA molecule, hence both the cells produced will be genetically identical. Let's move to a cell where the chromosome shape is relevant. Now, this question came in May, June 2016, variant 21. The diagram figure for 5.1 is a diagram of part of an animal cell in the stage of mitosis. You can see this is the centrioles. You can see the microtubule. The microtubule are holding the chromosome at the centromere. And you can see both the chromosomes have sister chromatids attached at the centromere. Next, this must be metaphase because metaphase is the only part of the cell cycle where this kind of structure actually is seen inside the cell. Draw a diagram in the space below to show the stage mitosis that follows in the stage of 5.1. So this must be anaphase. You will show the centrioles and you will see the shortened microtubules and you will show the microtubule attached to the centrioles and the centromere. So you need to show these three circles, the centrioles and the centromeres, and you need to show that the chromosome is moving in this way and the sister chromatids have been separated. So this sister chromatid is separate from the sister chromatid here and the same one in the chromosome above. You can see a few broken the microtubules here as well. So in this diagram, they are not asking you for any labels. So all the 3 marks are for a descriptive drawing. So the drawing must be elaborate and must show all these features. I hope that was helpful for you. That's it from my side. Thank you so much.